Hi everyone. I would like to thank organizing team to allow me to present my work. I, Dr. Vikram Dalall, today presenting study of anti-cancer effects of TTA-A2 and paclitaxel due to antagonistic interactions with T-type calcium channels. This work was already published, so I'm presenting it today. Where is it? Yeah. So this is like we have already published this work. So uh, ca cancer cells show a high proliferation rate due to various factors in the cells. One such factor is the increased calcium influx through T-type calcium channel, which maintains a continuous low influx of calcium into the cells. And in addition to that, calcium plays a vital role in the cells, such as cell division, cell signaling, hormone release, even gene expressions. So T-type blocking of T-type calcium channels already shown a decrease in the proliferations of various cancer cells like prostate cancer, breast cancers. So we thought that we aimed at blocking TTCC can also help to overcome the resistance in cancer cells towards anti-cancer drugs, such as calcium signaling plays a essential role in providing drug resistance for the organism. For that, we have used the T-type calcium channel A protein, and this protein it consists of two blockage sites. They were named as like port blocker. That's we mentioned is like just in short form PB, and the other one is like peptide gating site. That's we call it PG site. Yeah. So for that, we have as the structure is not available for that. We have predicted the three-dimensional structure, and the structure was refined and validated using the Ramachandran plot, Project, mole probability, and other web servers. Even like we have used the molecular dynamics also to predict the structure. So you can see that this this our structure has the calcium channel here, and we have predicted the structure. Further we have used the molecular docking to assess the binding of two drugs, that's TTA-A2 and paclitaxel. And we found that both the compounds have a good binding affinity at pore blocker and peptide gating site of this TTCC channels. As we can see clearly here that these both the drugs have interacted with the TTCC via hydrogen bonds and hydrophobic interactions, and they showed a good binding affinity with the TTCC. Yeah, you can see here both the TTA-A2 and PTX at the both sides, like pore blocking and peptide gating sites, have a good binding affinity. So further, we have further molecular dynamics was used to determine the stability of this protein with both the drugs. So we have performed the molecular dynamics in absence or presence of TTA-A2 and PTX at the both sides, and you can see that and we have analyzed the different analysis of the molecular dynamics, like root mean square deviations, radius of gyrations, hydrogen bonds. So RMSD, one of the analysis that is like RMSD results indicated that binding of TTA-A2 and PTX with TTCC forms stable TTCC ligand complexes, and TTCC pore blocking TTA-A2 complex is the most stable as compared to the other complexes. Further, like radius of gyration, we have determined the radius of gyration, that's like how much this protein remains compact after the interaction with these compounds. So we have our radius of gyration and SASA, that's solvent accessible surface area results, also confirm that this protein that's TTCC remains in stable conformation in binding of the TTA-A2 and PTX at the both sides. In addition to that, we have analyzed the hydrogen bonds results as well. That's we have analyzed the hydrogen bonding results in case of intraprotein hydrogen bonds as well as the intermolecular hydrogen bonds. That's the hydrogen bonds between the protein and the ligands. Both the hydrogen bonding results confirm that that's binding of TTA-A2 and PTX with TDCC form stable TDCC complexes at the both sides. And we came to know that the binding of TTA-A2 at PB site of TTC results in a higher stable complex as compared to the PG sites, that's peptide gating sites. Further, like we have analyzed the binding energy of the these ligands, that's TDCC and sorry, TTA and PTX with the TDCC. So there we came to know that that's both the ligands have a good binding affinity and they were stable at the both sides. Oh, there is, yeah. So overall, like I have not presented the other data that's like my other colleagues have done that's related to cell cultures and that. So overall, like from the study, we came to know that the binding of both the drugs, that's TDA2 and PDX with TDCC, can form the lower energy complexes. And this study indicated that both these drugs could interact with the off-target proteins as well through the less affinity when compared to its main target. If and the one more moreover, like this study indicated that drugs can also interact with the off-target protein in cells. And we came to know that if the drugs used in the treatment together, that's drugs here we have used the TDA2 and PDX, these can interact antagonistically and can be used sequentially treatment to a better choice without compromising the combination of the drugs. So I have presented not that much data, but just to give an idea like how we have done the things. I thought that's like to just give brief idea for this talk would be a good idea. So that's from my side. I would be very thankful. I'm very thankful to the organizer, and I would be very happy to answer the questions of any queries related to my talk.
Thank you very much. I was a bit curious. You mentioned that you used a homology model. For this. Can you describe a bit about how you generated it and what it was based on?
We actually the most homologous structure for this protein has a sequence similarity and identity of around like 50%, and we have used the Swiss model for that. Even we have predicted the model using the other web servers as well, like Modeler and Fiery tools, and all these web servers give us like similar results, and then we have validated those models using the different tools like Ramachandran plot, Project, even molecular dynamics we have used. So that's model was reliable, and then only we have proceeded with that.
Yeah, okay. We have another question about how you generated the grid for the docking.
Yeah, it's like the active site, as I have mentioned you, that there were two sites, like one is pore blocking sites. Generally there is like calcium binds, and the other one is it was reported in the homologous structures. That another one site they mention it's like peptide gating sites. That's you can just call it like allostric site. So we have kept our grid at the both sides. Even like we have performed the molecular docking using Autodoc Vina tools and even HatDoc as well. So that's like we want to like confirm that thing should not go in like bias. So like grid was like capped around the active site residues only. You can say that's calcium binding sites and peptide gating sites.
Okay, so I just wanted to ask, maybe I missed, but this data that you get from the docking, was that confirmed in an experimental setting by others, or are you still working on that?
We are still working on that. The thing is that that's other work like I have not presented here. That's what cell culture work.
Yeah, exactly. So you've seen the same effects in cells. Okay. I don't think we have any more questions at the moment, so I think we'll proceed to the next speaker and thank Vikram very much again for his
Thank you,
contribution.
sir. Thank you very much.
Thank you.